So if you clicked on this video, you're probably wondering about cell communication. Um, I wish that this was as simple as talking on a cell phone, which would only make sense since they're cells, but sadly it's not. It's a little more complicated than that. So I'm going to talk to you about just a few basic things. I'm going to start with the G protein pathway, um, then move on to the tyrosine kinase pathway. After that, we'll do ion channel receptors, and then intracellular receptors, and after that, secondary messengers. Um, just going to be a rough outline, really. Not going too far in depth, but far enough in depth to give you a slight understanding of how all this works. Um, I just hope this video helps, and if it does, make sure to hit that like button and subscribe. So, what the G protein pathway is, it's the most common pathway used in cells. Um, it's going to start out with a G protein linked receptor, which I'm going to draw right here. That's going to be attached into the cell membrane. I'm just going to roughly draw this with circles. But people say this works like a hand. I prefer to refer to it kind of as like a baseball glove, which that looks awful, but picture that as a baseball glove. So that's going to catch information from the outside and transmit it into what we call a G protein, which I'm going to draw right here. And that is going to get activated. It's going to be like an allosteric activation, and that's going to change the tertiary structure of this G protein through phosphorylation. So it's going to change the information that this G protein is going to transmit wherever it needs to transmit. Then this is going to go down the line, down the pathway, to wherever it is needed. That's going to start up GTP-ACE, which will in turn tell the cytoplasm, everything in the cytoplasm, the nucleus, what it needs to do, what it needs to change in order to fix the cell, what it needs to be making, stuff like that. This GTP, or this G protein rather, is a guanine nucleotide. Just throwing that in there. Alright, next up on our list is the tyrosine kinase pathway. I like to call this a fire alarm, almost. It's mainly used in trying to fix and heal the cell, but it can also be used to stop things like cancer, try to prevent those. What it does is it activates 6 relay proteins at once. So if the cell is damaged, or if the cell is dying, this is going to occur. This is going to activate those 6 proteins, and it's going to prevent the cell from dying, or at least attempt to prevent any permanent damage to the cell. This is a very important part. So the next pathway, it's not really a pathway, but the next thing we're going to talk about is the ion channel receptor. And what this is going to do is it's going to accept in these molecules out here, which usually it's only going to accept one of them in, one type of them in. So if it's accepting blues, those blues are going to come in here. If it's accepting reds, those are going to come in here. But if not, those are going to have to move somewhere else to be brought in by another ion channel receptor. This is kind of like a funnel into the cell. It's also kind of like a gate. So those are going to enter through here and go into the cell, whereas these other ones are going to have to go elsewhere to be entered in. These ion channel receptors, they have to be activated. It's not activated the same way a G protein is. It's activated a little different. So what happens here is a ligand, or a neurotransmitter, which they're also called, is going to activate this and tell it to let these things in, and if it's not activated, neither of these are coming in here, and both of them will have to go to an activated one. Next up, which is similar, is intracellular get that corrected. Intracellular receptors, and these are a little different than any of the ones we've talked about so far. They don't attach to the cell membrane. So I'm going to draw a cell membrane right here. Um, it's really rough. That's not at all what it looks like, but there you go. Anyway, so these are going to be here in the cytoplasm. I'm just going to draw it like that. That's not at all what it looks like. Don't at me on that. So draw the, this is going to be the nucleus. It's rough drawing. Anyway, it's going to be here near the nucleus. And what it's going to do is hormones and steroids from outside the cell, this is going to be your hormone and your steroid, are going to come in. They're going to diffuse into the cell. And this, the intracellular receptor, is going to be connected to a ligand that can receive hormones and steroids, because this alone cannot. So these things are going to come in and attach to this, and this guy is going to tell the nucleus or stuff in the cytoplasm what to do, based on these hormones and these steroids coming into the cell. So this is meant to boost or decrease a gene's transcription. And of course, the main thing to remember here, these are not on the cell membrane. The last thing we're going to talk about is another group entirely of messengers. That's going to be cyclic AMP and IP3 slash calcium 2+. So first we're going to talk about that cyclic AMP. So cyclic AMP is going to start at the same place as the G protein pathway. So it's going to receive that information. It's going to activate that G protein right here. And when that G protein is activated, it's also going to activate a process known as adenylyl cyclase, which is going to make this CAMP. Now, what CAMP does, once it's created, it is like a megaphone for that G protein. It's going to magnify the message that it's going to make. It's going to make that, in a way, louder to the rest of the cell, so the rest of the cell can get it quicker, and the processes that it needs done can start quicker. And it can do both of these things. It can change molecular activities in the cytoplasm, or it can start a new pattern of gene transcription. So this is pretty important. So the last thing I'm going to talk about in this video is going to be IP3, which is inositol triphosphate, and then calmodulin, which is Ca2+, and that is literally just a calcium ion. That's all that is. But these two are going to work together. More like this one's going to work to create this one. So IP3 is essentially, it leads to the creation of Ca2+. So what's going to happen is IP3 is going to, this is a really rough drawing here, but this is the endoplasmic reticulum. IP3 is going to, that's the IP3 right there, it's going to attach itself to the endoplasmic reticulum, and in turn, the endoplasmic reticulum is going to release those calcium ions, which are the most common or most widely used messengers in the cell. I'm going to draw those as these little dots down here. And what those are going to do, those are going to cause muscle contraction. They'll cause exocytosis, adhesion of cells to the extracellular matrix, and many more things. The list just keeps going on and on. But this process is extremely important. It helps us to move. That's pretty much it for this process. Alright, anyway guys, thank you for watching this video. Hopefully this helped you. And if you did enjoy it, make sure to hit that like button, subscribe. Maybe I'll come out with more videos. Depends on how this one does. But thank you guys. See you later.